In this study, we have evaluated the patients with smoldering myeloma, and we have tried to highlight those who may have a good risk. Now, what we have is a very unique set of samples. We have patients who have not progressed from smoldering myeloma to myeloma for 5 years or more. And then we have patients, 31 patients, who progressed, and we have paired sample collected when they had smoldering disease or MGUS, and then we have collected their sample when they became myeloma. And then we have done high throughput genomics, both whole genome sequencing as well as RNA sequencing, and we have tried to analyze what predicts the non-progressor group, that the patients do not progress. And in this abstract, we have identified that there are certain mutations, there are certain significant differences which are observed in the coding regions. So if you look at patients in MGUS stage, there are the number of mutations are lower compared to myeloma and compared to the progressors group. We observed very low NRAS, only 3%, and BRAF, 3% mutation frequencies in non-progressor smoldering myeloma, and that's an important finding. And we did not observe driver mutation in FEM46, TTN, cyclin D, etc., in this patient population. So one was that we had differences in mutation pattern that predicts for outcome and results. We also looked at what we described recently in our paper in Journal of Clinical Oncology, what is called genome scar score. And genome scar score is low in the non-progressor smoldering myeloma, it was 3, compared to progressive myeloma, which was 11. And so genome scar score probably would help identify patients who are likely to remain smoldering in a good risk group compared to other. And then we have done the RNA sequencing analysis to see are there any specific patterns that may predict outcome. And we are beginning to describe some of this pattern. I think the progressors patients already have acquired certain signatures of aggressiveness, you would say, that may be enriched in MYC targets, DNA repair gene, and mTOR pathway, and that may predict the progressors from non-progressors. We have also seen that certain signatures of mutations, such as ApoBec, are also important in this patient population. So to summarize, in this abstract, we have defined the differences between smoldering patients who are not progressing for long period, 5 years and more, compared to those who have progressed, and identified the mutational patterns. We have identified the signatures of the mutations, and we have looked at expression profile and pathway activation that may differentiate this group. And so this is one of the first such effort that we begin to develop a genomic-based algorithm for identifying good risk patients, and then the rest could be considered higher risk patient. Venetoclax, as you all know, is an important drug that targets BCL2. It's been approved for other malignancies, lymphoid malignancies, but in myeloma, we have evaluated and found it to be effective, especially in combination with bortezomib. It's exceedingly effective in patients who have T1114 translocation, where cyclin D1 is upregulated, but BCL2 is upregulated. And this is where it is being utilized. However, patients do relapse, and we are beginning to study the markers for resistance to Venetoclax. So in our study, what we have done is that we first looked at what we have called extrinsic resistance, where we took the 2 cell lines, treated with Venetoclax, and induced resistance over time, and then identified individual clones. And what we find in this study is that the resistance process appears to be heterogeneous, and I think that is important to keep in mind. And we have looked at various other pathway and identified that ERK pathway may be activated in the resistant clones that we have identified, and the ERK inhibitor leads to addition to killing. Then we try to correlate with intrinsic pathway, inherent pathway that may be activated. And we find that the cell lines with intrinsic resistance do not have a very similar pattern as what we found in extrinsic resistance. ERK was not super significant in this group. What we find, which is well known, is that high MCL1, high BCL-XL are the drivers, change in BCL-XL and change in MCL1 are driver. And so the ratio BCL2 to BCL-XL or BCL2 to MCL1 is high, not the two genes are high. And this has been evaluated in other malignancy as well, and that's what we find in myeloma. We have identified and we report that one of the gene, GOS2, is decreased and may be important for intrinsic resistance, and we need to understand its role further as we move forward. So we have provided evidence in this abstract of the molecular mechanisms of acquired resistance to Venetoclax with activation of ERK pathway as one of the prime target. And that now will begin to give us molecules that can be combined with Venetoclax, such as ERK inhibitor on one hand, but also considering BCL, MCL1 at least targeting molecule, which are already in the clinic for potential synergistic activity. BCMA CAR T cells are very exciting. They are, in this ASH meeting, there are many studies being presented by various groups, United States, Europe, and China, with various CAR T products targeting BCMA. There are studies from larger data, Karma study and Johnson & Johnson Legend study. And then there are other investigators who have presented, and Gang and others, to show how BCMA targeting CAR T cells works. Almost all of these studies have a very similar data, which is that response rates are very high, 75 to 95%. The toxicity is limited. The main toxicity ends up being cytopenia because of mainly lymphodepletion. The CRS, cytokine release syndrome, happens in almost everybody, but its severity is lower. It's grade 1 or 2, very rarely. 5 to 7% could be grade 3. So there is a tocilizumab used in 50% or so, but it is limited compared to other malignancies. And very importantly, different from lymphoma, neurotoxicity is not the commonest issue with these patients. So we have to keep that in mind. So the concern still remains is about the PFS, progression-free survival. Even though responses are very high and they are deep, half of the patients who get CR or VGPR have MRD negativity, and in some studies even higher. However, PFS ends up being around 8 to 12 months, so patients still relapse. So now there is a great excitement for this drug because patients being treated in these studies are very late. They are 5th and 6th line treatment and beyond. And even then we get such a good response. So now the effort is, how do we improve the PFS? And many new things are being planned, and some of them are already in the clinic, which is utilizing CAR T cell earlier on, second line, and in some cases even high-risk patient first line. Also come up with strategy to prolong PFS by having some what we would call maintenance treatment. There are studies now being shown where 2 CAR T cells are being infused at a certain period of time, or they are combined with other immune modulating agents and other immune stimulants. And so there's a great excitement about CAR-T with its efficacy. And now we are beginning to use it at an early stage. And stay tuned to have more data that brings it to the clinic and to the patient in a very near future. In this ASH meeting, there are the whole focus. If I have to summarize the meeting, immune therapy has been the greatest focus so far. Immune therapy in multiple ways, not just immune therapy as in CAR-T, just the way I described, but immune therapy in the form of utilizing different targets, and immune therapy utilizing um, uh, various other mechanisms, including bispecific molecules. So we have to keep in mind all these various newer treatment options which are becoming available. So if I give an example, BCMA is the most wonderful target. And I think we have to keep that in mind as we move forward. But besides BCMA, there are two other targets which are being analyzed in the current stage. So one of the target being analyzed is GPRC5D. GPRC5D is a very exciting target because it's expressed predominantly on myeloma cells. And in the early clinical study using bispecific, there is a very good response with really minimal toxicity. And so GPRC5D is an important new target for CAR-T and for bispecifics. Then the second thing we have to keep in mind is there is another target called FCRL5. That also is expressed predominantly on myeloma cells, could be on 1Q, could be subtype specific. And in the early results in bispecifics, it also shows a good response. So we have two new targets besides BCMA for immune targeting. The second component, CAR-Ts are very effective, but there was good data presented using BCMA targeting as well as this new targets connected bispecific antibodies. And so the bispecific shows very good efficacy, good tolerability with limited toxicity. They do get CRS, but there's a limited toxicity. So I think the most exciting part of this ASH meeting was the immune-based therapy, the efficacy of CAR-T, which is moving forward, and the BCMA and other novel target antigen targeting bispecific antibody, which are off the shelf, which can be given to the patient and provide deep and durable response in this patient population. So in this ASH meeting, I think immunotherapy for myeloma has been the most important, and these antibody-based therapies are of great significance and great interest to us in coming years.